moving towards our last speaker. Our final speaker is Bradley Dearnley from University of Sheffield in the UK, presenting a stretching and squeezing of neuronal log firing rate distribution by psychedelic and intrinsic brain state transitions. So, Bradley, go ahead.
Thank you very much. Okay, so yeah, I'll be speaking about the stretching and squeezing of neuronal log firing rate distribution by psychedelics and intrinsic brain state transitions. So understanding the psychedelic effects has been quite hard due to the restricted status of some of these compounds, in particular serotonergic hallucinogens. So although we know at the cellular level they target the 5-HT2A receptor to exert their effects, and recently some of the few authorized studies have shown us that there's some functional connectivity changes under the influence of these drugs. The microcircuit level remains relatively understudied despite being critical to understand the mechanism of action of these drugs. So we use Neuropixels probes to record from large neuronal populations in mouse medial prefrontal cortex. So we know that the expression of the 5-HT2A receptor is dense in the medial prefrontal cortex, and these Neuropixels probes allow us to record even an order of magnitude higher number of well-isolated neurons than previous work. So we can get 300 or 400 well-isolated neurons per recording to really probe what's going on at the microcircuit level when we look at use these drugs as well. So we administered our drug orally. So the drug that we used was TCB2, which is a high affinity, potent 5-HT2A receptor agonist. We trained our mice to drink a sucrose and bitterant solution so that when we replaced it with TCB2, they'd be happy to drink a sufficient dose. And it also reduced any confounds of injecting the mice in the neural activity that we might see. So not only does it emulate the human administration route, but it also stresses the animals less. So how do we know that it works specifically with TCB2, the drug that we used? Well, we saw our mice exhibit head twitch responses, which is a robust indicator of hallucinogen action, specifically at the 5-HT2A receptor. So you should be able to see head twitch here from one of our mice. There we go, after it had been given TCB2. And then we tested as well the presence of head twitches. So we did this in two ways. So the first on the left, you can see is a between subject design. So one animal was given a control bitter solution and the other the drug, and the observer scored the head twitches. This was about 20 minutes after dosing. And you can see that the observer was able to reliably distinguish at levels significantly above chance which animal had had the drug based solely on head twitches. And this was the case as well for when we used a control and TCB2 in the same animal 1 day apart. We can also see that there was the experimenter observer could reliably distinguish between the two animals solely based on head twitches observed. So our design, we recorded just baseline activity of a head fixed mouse, and then the design was a within subject design. So the animals were given both a control and TCB2 on 2 consecutive days, with the order being randomized, and they drank the solution for around 15 minutes before we then recorded activity after drinking for around 45 minutes. So the first thing that we looked at was the LFP power. And when we look on the left plot here, we just looked at in the beta power range, and at the level of individual recordings, we can see that there was a quite pronounced suppression of beta power from before to after drinking in the TCB2 conditions, which are significantly different to controls, which didn't change much at all. And then on the right, you can see that the ratio from before to after LFP power in several frequency bands, you can see it's actually quite widespread. It even starts from below the gamma range right up to into the low below the beta range, right up into the low gamma range. So next we looked at firing rates. So this is just an example of single neurons on long time scales and how variable they are. This is one of our neurons, and what we did to quantify the long-term changes was to fit a step function. So the best fit step function, it provides us with the time position of the best fit step, and also how large the best fit change in the firing rate over the course of the entire recording was. So looking at these changes in firing rate, in red you can see the drug, and in black control. Each point here is a neuron, so this is over 3,000 neurons from several recordings all pulled together relative to the point of drinking, and you can see the timing on the x-axis of the fitted step regression, and on the y-axis the change in the firing rate, the size of the step. So we looked for this plot, when we looked at the density of the step timing, we took only those units that had a greater than twofold change in their firing to make sure we were only looking at those neurons which really changed significantly. And what we see is this really big peak at the onset of drinking, which we kind of expect, and that was present in both control and TCB2 recordings. But then we also see this really prominent second peak, which is only present in the TCB2 recordings, which may be attributed to the drug, and it's about 12 minutes after the onset of drinking. And then we looked again at just those units that had a greater than twofold change in their firing, but split between those that increased their firing by a lot and decreased their firing by a lot. And we find that in both cases, there's a significantly larger portion of significantly modulated cells in the TCB2 condition compared to controls, but that this effect is much more pronounced in those cells that are decreasing their firing rate in response to the drug. So next we actually looked at the magnitude of the changes in firing rate with the same color scheme here, red for the drug and black for control recordings, with the firing rate before and after plotted on the X and Y axis. And when we look at the difference between the two, we can see that this deficit in the middle here corresponds to the control neurons being fairly close to the diagonal, whereas the surplus in yellow shows us that the TCB2 distribution, the TCB neurons are much more modulated in their firing rate by the drug than drinking the control solution. And as you can see here, most of the surplus lies below the diagonal as well. As we saw before, most cells show a significant decrease in their firing rate as opposed to increase. So to quantify this, we used the absolute rate modulation, which is just simply the log rate after divided by log rate before. And when we look at the distribution, we can see that there's a high proportion of cells in the control recordings which show a low absolute rate modulation, whereas in the TCB2, the density of the distribution is pushed up further into the higher rate modulation. So we also found that TCB2 stretches the log firing rate distribution rather than just changing the firing rates. It actually altered the distribution. So in the left plot, you can see here, this is just the after portion, but the pooled distribution from the TCB2 after drinking and the control after drinking distribution. And you can see that tail is stretched towards the left, showing that the large proportion of cells that significantly decreased their firing rate. But on the right, we show an analytical fit, and you can see the log rate distribution shows a really significant stretch when it moved into the psychedelic brain state, so from before to after drinking in only the TCB2 recordings. So it's also been shown that these log rate stretching, squeezing is present in other brain state transitions. So for example, from the transition from sleep to wakefulness, which led us to ask the question, is it a general phenomenon rather than just specific to the psychedelic brain state? And to address this question, we looked at the pupil indexed arousal. So in all of our recordings, we also measured the pupil area. So on top, you can see the pupil area traced throughout the entirety of a recording. We classified high and low arousal portions of the recording as those parts where the pupil area was in the top quartile and the bottom quartile as high and low arousal respectively. So we ran exactly the same analysis on low and high arousal states, and we found the same thing, that the log rate distribution when the network went from a low arousal state to a high arousal state, that we saw the log rate distribution was indeed also stretched. So just to confirm that our effect of TCB wasn't completely explained by arousal, we also looked at the change in the pupil size in the drug recordings as well. and we found that there was no change in either the control or TCB2 recordings from the before to after portion, and there was also no difference between the control and TCB2 pupil area ratio change. So then we next split these cells that we'd recorded into those that upregulated their firing rate and those that downregulated their firing rate in response to the drug. And you can see here the dashed lines refer to the before drinking baseline state and the dotted lines to the psychedelic post-drinking state. And we see that in the before state, the downregulated and upregulated cells have a very similar baseline firing rate. And then when the downregulated cells move to the left, their neuronal distribution is stretched, log rate distribution, is stretched. And the cells that increase their firing rate their distribution is also squeezed. So we see stretching and squeezing dynamics actually even within these subpopulations, which then when you look at the whole distribution leads to just a global stretching of the distribution in the psychedelic brain state compared to controls. And this was the case because there was a very strong correlation between the baseline firing rate and the rate modulation. So in the orange upregulated cells here, you can see a very strong negative correlation when we look at the baseline firing rate and also the rate modulation by the drug. And the opposite is the case in the cells that were downregulated. But what we actually see here is in both cases, the rate modulation is tending towards zero as the baseline log rate increases. So if we look in the black here, you can see it's the running median. The absolute rate modulation was much greater in those cells which were slow firing and much lower in the fast firing cells. So to summarize, thank you. We established an oral administration protocol for hallucinogenic drugs to allow stable acute recordings and also to emulate human administration. We replicated the low frequency power reduction, which has also been seen with other typical serotonergic hallucinogens such as LSD and psilocybin, also in humans, not only rodents, as well as the head twitch response as well, just to verify that our TCB2 was working as we anticipated. When we looked at the firing rates, we saw that TCB2 caused a significant modulation of firing rates in the medial prefrontal cortex, but there was a much greater tendency towards suppression of firing. And then when we looked at the log rate distribution as a whole, we found that going from a baseline to a psychedelic state or going from a low arousal to a high arousal state was accompanied by an increase in the standard deviation of the log rate distribution. And we propose that it's a generic framework for other brain state transitions as well, not just the psychedelic brain state. And this stretching of the log rate distribution is explained by two bidirectionally modulated subpopulations, with the upregulated population also being squeezed in itself and the downregulated subpopulation also being stretched. And this is due to the much greater effect of the drug in slow firing neurons compared to fast firing neurons. And this is our recent preprint, and thank you to all of the authors on the paper and PI, Michael Oken as well. So thanks very much.
Awesome, thank you. Thank you so much, Bradley. Amazing talk. So if in the audience there is anybody having questions, please do ask. I have one question that is like, I've seen like from the one of the very first result slides you showed, like you were showing like this reduction in beta, like suppression in beta. Did you look at other frequency bands as well?
Yeah, so it was quite widespread, even below the beta and even slightly above. So we saw kind of, if you go like right up into the high gamma range, then the effect is not there. And even sometimes you find that the power is increased in the high gamma range. But most of the low frequency power are kind of all the way up from like alpha right up to low gamma. You see this suppression, but most pronounced in beta. Yeah, the beta range tends to be highly suppressed. Yeah.
Awesome. Thank you. All right. So if there are no other questions, I would like to thank all our